வணக்கம். வெல்கம் டு NCERT Biology Reading. NCERT Biology Class 11 Chapter 10 Cell Cycle and Cell Division. 10.4 Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half result in the production of haploid daughter cells. This kind of division is called production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come we come across meiosis during gametogenesis. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cell of DNA replication. Meiosis 1 is initiated. Meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and recombination between them. Four haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped under the following phases. Meiosis 1 contain prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis 2 contains prophase 2, metaphase 2, anaphase 2, and telophase 2. 10.4.1 Meiosis 1. Prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior, that is, leptotin, zygotin, pachytin, diplotin, and diakinesis. During leptotin stage, the chromosomes become gradually visible under the light microscope. The compaction of chromosomes constituted through leptotin. This is followed by the second stage of prophase 1 called the zygotin. Prophase 1 called zygotin. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate the chromosome synapsis is accompanied by the formation of complex structures called synaptonemal complex. The complex formed by a pair of synapses homologous chromosome is called the bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of the prophase 1 are relatively short-lived compared to the next stage, that is pachytin. During this stage, bivalent chromosomes now clearly appear as tetrads. This stage is characterized by the appearance of recombination nodules, the site at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over leads to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of pachytin, leaving the chromosomes linked at the sites of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombinant homologous chromosomes of the bivalent to separate from each other except at the site of crossovers. These X-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiotic prophase 1 is diakinesis. This is marked by terminalization of chiasmata. During this phase, the chromosome are fully condensed and the mitotic spindle is the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of dikinesis, the nucleus disappears and the nuclear envelope also breaks down. Dikinesis represents transition to metaphase. Metaphase 1, the bivalent chromosomes align on the equatorial plate, figure 10.3, the microtubules from the opposite poles of the spindle attach to the pair of homologous chromosomes. Anaphase 1, the homologous chromosomes separate while sister chromatids remain associated at their centromeres. Chromatids remain associated at the centromeres, figure 10.3. Telophase 1. The nuclear membrane and nucleolus reappear. Cytokinesis follows and this is called as dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extent state of the interphase nucleus. The stage between the two meiotic division is called the interkinesis and is generally short-lived. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. 10.4.2 meiosis 2. Prophase 2 meiosis is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2. At this stage of the chromosome align at the equatorial and the microtubules from opposite poles of the spindle get attached to the kinetochore. Figure 10.4 of sister chromatids. Anaphase 2. It begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move toward opposite poles of the cell. Telophase 2. Meiosis ends with telophase. Telophase 2. Meiosis ends with telophase, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is, four haploid daughter cells. 10.5 Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved by across generation is sexually reproducing organisms. Even though the process per se paradoxically results in reduction of chromosome number by half, it also increases the genetic variability in population of organisms from one generation to the next. Variation are very important for the process of evolution. Thank you.